The I See Dead Plants podcast shares the stories of people and plants, pests and pathogens, and the conflicts among them. Join us as we speak to the folks who attempt to solve complex problems with plants through pest management research. I See Dead Plants is an extension-based podcast created by the Crop Protection Network and the North Central Integrated Pest Management Center. Music written and recorded by fellow plant pathologist Sean Toporek and hosted by Ed Zaworski.
You're tuning in to part 2 of my discussion with Dr. Will Rutter of the USDA ARS, and we're discussing root knot nematode and the effects it has on vegetable crops and what we've learned over the years. So thanks for listening, folks. Enjoy part 2. One of the other things that I learned and thought was interesting about the root knot nematode in reading the review is you were mentioning like they're mitotically parthenogenic. And so it's a little confusing, right? Like how they I guess maybe for the listeners, I read the paper. If they don't read the paper, do you want to explain like mitotic parthenogenesis and like, I guess how that leads to a little bit of confusion with how these things are overcoming genetic resistance to their hosts and that kind of stuff. I thought this was really cool stuff.
Right. So when we talk about a parthenogenic organism, we usually talk about something that's asexual. So they don't require sex. And in general, we think of sex in animals as a way to diversify your offspring and make them more plastic, more able to adapt to their environment. And so there's a lot of different parthenogenic organisms. And there's a bunch of different parthenogenetic, but they're parthenogenic in different ways. So when we say a mitotically parthenogenic, that means they're basically clones of one another. In a general sense, they are basically taking their genome unchanged, and they've confirmed this, by resequencing multiple genomes. They pass their genomes almost completely identically down through the generations, and that it's happened so long now that if you look at their chromosomes, they don't look like, when you think about human chromosomes, we have these nice, pretty what are called bivalent chromosomes. You see the sister chromatids, they look like little X shapes, and those are the sister chromatids together. But in these mitotically parthenogenic root knot nematodes, they don't have that. It's just fragments of chromosomes, all just a mess. It's hard to even count them. They estimate there's somewhere between 44 and 47 chromosomes or fragments of DNA, but they're not being mixed up or going through meiosis like a normal sexual organism is. Why that's odd is because we know that these nematodes, like I said, have these huge host ranges. And so they have to avoid the plant immune system, just like all plant pathogens do. Mm-hmm. And they do so very effectively. So if you think in terms of host resistance, probably the most well-characterized source of resistance to the southern root knot nematode is in tomato. And it's referred to as the MI 1.2 gene. So it was deployed in tomato several decades ago, and has since been put into most commercial tomato varieties that are grown in the southern U.S. But the southern root knot nematode has repeatedly shown that it can basically infects those resistant plants enough, it can overcome that source of resistance. And so it's been a question for a long time how they're able to change their genome in the absence of sex if they're a clonal organism. And it's clear from what we know from other pathosystems and what we know about the MI 1.2 gene. We know it's what's called a standard or a canonical nucleotide binding leucine-rich repeat type protein. So it's an NBSLR for short. Okay. And basically, we know that these NBSLRs, in the context of plant pathogen interactions, basically detect the presence of an effector protein within the cell, either directly or indirectly, and then trigger that plant cell to kill itself.
Mm-hmm.
And when it does that, because you're talking about a biotrophic pathogen that requires a living host, you're essentially killing that one cell, but you're also killing the pathogen itself.
Right.
So that's the idea for the plant point of view.
Mm-hmm.
So in other plant pathogens, we know that an avirulent pathogen, ones that has the cognate effector that's targeted by that NBSLR, if it's able to lose that effector, well, then it becomes virulent, because that trigger is no longer able to detect that individual protein, and therefore is no longer able to trigger host defenses. But when we look back at root knot nematodes, well, how is it losing or perhaps hiding that effector protein if it's not able to lose genes, or if it's not changing its genome at all? Although I don't think we have a complete answer. It's certainly clear that the genomes do change, just not in the way we would think of in a sexual organism, right?
Right.
It's still losing genes. The genomes are still rearranging. That causes and those changes are being caused and leading to those new virulent populations, but it's just happening via a mechanism that we don't fully understand yet. Yeah.
Right. This has me, you mentioned, I did a podcast. If you haven't listened to it, folks, I did a podcast with Horacio Nakora Lopez and Emil Glucktaler. Man, it has me thinking about that, the starships and the giant massive mobile elements. Like
exactly. And we've thought about similar mechanisms in the root knot nematode genome as well. Yeah, it's been well documented that transposons and mobile DNA elements have a significant impact on pathogenesis in fungal plant pathogens. So it's not too much of a stretch to think that there could be a similar mechanism in nematodes that just really hasn't been discovered or characterized yet. Yeah, because there are certainly families of transposable elements that we know are in that genome and could be having those types of effect on the genome. Yeah.
So as of like right now, I mean, you mentioned like the effector proteins being a thing that maybe we can learn more about with the advancements we've made in mapping these genomes, essentially, right? So is that the bulk of the work that's going on right now, is just trying to identify different effector proteins and what they do, more or less?
I mean, there's different labs looking at a bunch of different aspects of nematode pathogenesis. My background, because it's in plant resistance, and I did my PhD on nematode effectors, so I'm a little biased. I always think that's the best avenue to pursue. But I think it is kind of developing into a new and useful avenue. We've been saying it for a long time that understanding these effectors could provide useful, resistance tool in the future. And I think we're just getting to the point where that might be the case. That we can really see the functional outcomes. I can give you an example. If you know that a effector targets a specific region in the pathogen genome or a specific protein within the host genome, and you could use a approach like CRISPR-Cas9 and go in and alter that target so that effector no longer interacts with it. Well, now you've just been able to use that information to design resistance. So right. I think we're just now starting, in the biotechnology realm, seeing the possibilities that could arise from that. So I think we're building the case more and more, I think, for that we need more understanding of what these proteins are doing.
Yeah.
And understanding, how we could use them to further plant breeding and resistance breeding in particular. Yeah.
Yeah, man, I'm just thinking, if you're a young person listening to this podcast, there's probably not that many of you, but no, but if you are, this is such an exciting field, an exciting area of study. And any plant pathologist that you ask about how do you best manage a pathogen, especially nematodes, right? Like they're difficult to manage with chemicals. They're difficult to manage with some of the cultural practices, but like genetics, host resistance and genetics, it seems like the best way to do it. And it seems like we're cracking the surface now. I say we're, people like you I'm not going to lump myself in there, but people are just cracking the surface on this, and there's lots of avenues to go down, as you mentioned, whether you're talking effectors or some other avenue. It's exciting stuff. I get really pumped up when I talk with people about this stuff. So
yeah, and there's there's new traits coming out, biotechnology traits. I think about Nemasphere. What's the new BT trait coming out?
Oh, I'm not
for
familiar.
soybean cyst nematode. I'm sure there'll be other targeted traits coming out again for different nematode species in the future as well. So I think that's just the beginning. Yeah.
Gotcha. Well, yeah, I mean, that was I got one more for you before I move on. I in reading kind of like your summary points at the end of it, and this is just clarification for me. I couldn't get my brain to figure this out. You mentioned like that one of the things that could be worked on with regards to learning more about root knot nematode would be to increase the number of methods for transforming root knot nematode.
Yeah. So
I guess if you go and read the paper, which I recommend you do, it's in there. But yeah, what was meant by that? Just for
right.
For my own
So transformation is simply the method by which we put basically exogenous either DNA and or protein into a cell to alter its genome. So you think about that in terms of how they made Roundup, like how they came up, they developed the Roundup Ready trait.
Sure.
in soybean. How they come up with new biotech traits is through the process of in order to do that, you need a transformation system, some way to get the molecular tools that alter the genome actually into the plant and have it create a change within that genome that is then passed on to that organism, whether it's a plant or an animal, that offspring.
Gotcha.
So that's referred to as transformation. So, and right now, root knot nematodes and cyst nematodes in general are a recalcitrant species. There's no real way that I've heard of yet that we can reliably transform them or make any changes to their genome. So we don't have access to a cool tool like CRISPR-Cas9, that can target. We can do that if any species that can be transformed can potentially be targeted. We can target specific genes using CRISPR-Cas9. But right now, if we want to understand how a specific gene works or how genes work together in the nematode, we're limited by what we can do indirectly through its host. So one of the ways we go about that, we either overexpress the gene in the plant and see how it affects nematode pathogenesis, or we target it through a process called RNAi, where we can make the nematode ingest double-stranded RNA and knock down a specific gene.
Sure.
But if we could, take and actually alter a gene or put a new gene into the nematode or a tag gene into the nematode, we could really start to better understand how that effector gene functions in pathogenesis.
Do you need to keep up to date on new pests or pests that may be moving into your area? Pest Alerts focus on new pests, pathogens, and weeds that are popping up in the North Central region. Check out the North Central IPM Center's Pest Alerts page and stay a step ahead.
So is that just mostly because of the complicated nature of the genome for root knot that it's hard to do transformations? Or
I mean, there's a lot of barriers, certainly. So, you think about the model system for nematodes is C. elegans,
Hmm.
and the way they do it for C. elegans is they take like a gravid female and they have this array of pins that they soak in whatever they're trying to put into their genome.
Uh-huh.
And basically they run the gravid females over these pins to get the constructs that they're interested in into the nematode. And then they have what's called a marker. So I think it varies, but they basically are able to mark which ones have actually been the offspring that have actually been successfully transformed.
Oh, okay,
And
okay.
so you're actually able to select the ones that have developed the trait or lost the gene that
Hmm.
you're interested in. Yeah, we don't have anything like that. I know people, different labs have tried. I'm hopeful in the future, that may we'll be able to develop something,
Right.
that'll be useful. But who knows? I think it would definitely mark a turning point in nematode analysis, right? Nematode molecular analysis if we were able to do that. Yeah.
Yeah, it's exciting stuff. Definitely recommend reading the paper, folks. There's one more thing I wanted to talk to you before we get into wrap up questions, Will. And it's as if, things weren't sophisticated enough or complicated enough. There's a new species of root knot nematode that's kind of coming onto the scene in the southern United States, right?
Yeah, yeah. So, like I said, we have the Meloidicon incognita group that have been around for 50 to 100 years. It's hard to know how long, but those have been around so long that we've developed these types of resistant crops, with resistance genes that could be deployed to help manage those species. But more recently, there's been an introduction in the past 20 years of this, what's commonly called now the guava root knot nematode, Meloidicon interlobii. And it's come in primarily through the movement of nursery crops likely, and the movement of machinery between farms. Anything that moves soil or living plant material can move a root knot nematode very effectively. And so it's kind of been slowly making its way across the southeast. It's now in the majority of counties in Florida. It's been found. It has a very wide host range, just like the incognita group, but it's just different enough that it pretty much ignores all of the host resistance,
Ah,
that is effective against those endemic species. And so
okay.
it's a serious problem. Yeah, yeah.
So that's kind of what accounts for, like it just in my, I don't know, whatever you want to say, some skim reading I did about it's enterolobii, right?
Yeah, enterolobi. I've heard it pronounced both ways. I'm not a linguist. So, yeah,
You
I
got to
won't
say it
like.
loud and proud, right? In Latin. uh, essentially, what I've read is it's a more virulent nematode, but is that essentially just because it's different and it's not
It's
there's no
it's
host
hard,
resistance to it?
it is a very thermophilic species as well. So in high heat, it can reproduce a lot faster than incognita group species. So it has a very, and then it also interacts with different hosts a little differently. So we can make very large galls on specific hosts.
Oh, gotcha.
You know, but also again, it's kind of a host specific, even host variety dependent interaction.
So you said it's kind of slowly creeping along. Where has enterolobi or enterolobii been found thus far?
Again, most counties in Florida it's
Florida.
been found in. After that, it was found. It got into the sweet potato industry in
Hmm.
North Carolina, so it's
Okay.
now in the majority of sweet potato growing counties in North Carolina, and that's incited several states to impose quarantines on North Carolina for sweet potato movement, and South Carolina as well. So we found it in two counties in South Carolina, as well as two counties in Georgia as well now
Gotcha.
too. Now, how long it's been there, I think that's up for debate.
Hmm.
Again, it requires a molecular test. to really distinguish the different species. And there's so much root knot nematode in these areas already.
Right.
The reason the sweet potato growers noticed it because it started causing a lot of damage on previously cultivars that were previously thought to be resistant to the incognita group, and now they're getting severely damaged and a lot of yield loss in those cultivars. A lot of the work I do now is on Antilobii, identifying new source of resistance in sweet potato, in pepper, cucumber, and watermelon, and trying to get those into lines for release that could be used to help develop new resistant varieties of these crops to help manage this nematode.
Since resistance isn't as viable of an option of management of these, how does the management differ for Interlobii in the time being? Are people just using a lot of like nematicides, seed treatments, in-furrow stuff?
Yeah, there are a lot of nematicides that are still effective against Interlobii. We have again, my colleague Johan Desiger has done a lot of work on cover crops, the effective cover crops or different crop rotations. Peanut so far has been a pretty solid non-host, but there's other poor host or non-host crops that could be effective rotational options. So
Gotcha.
it is causing some growers to have to rethink their rotation schemes
Gotcha.
and their nematicide applications. Yeah,
makes sense. Well, I've grilled you a bunch, and I'll grill you a little bit more with some kind of wrap-up questions here, but I like to always pause at this point and see if there's anything that we didn't cover that you wanted to cover with regards to root knot nematode or Meloidogyne Interlobii or anything that we've talked about today.
Yeah, that's it. The only thing, I talk more about, Meloidogyne Interlobii, the best way to manage is try not to get it in the first place. Ideally, that can be avoided by purchasing certified seed nursery stocks.
Gotcha.
So the movement is largely in sweet potato has largely been vectored through the movement of infected seed potatoes. So if you're not familiar with sweet potato, it's a clonally propagated crop. So every few years, growers buy new quote unquote seed roots that they plant in the soil and then grow out to create slips, cuttings that are then used to plant their production beds.
Interesting.
So if one of those seed roots is infected with Interlobii, now that entire field is infested.
Yeah.
So
that is interesting.
It becomes serious. Yeah. And it's not just sweet potato, obviously they have to look at their roots, but I think it goes back to the importance of nursery, clean nursery stocks in general, especially since this nematode we found it in spots that have no history of sweet potato production. And like it was likely brought there through the movement of a different nursery stock that was infested. So clean nursery stocks is a key to kind of slowing this nematode down until we have some better, management tools for it. Yeah.
Are there entities that are inspecting nursery stocks then? Like, I guess.
Certainly in different states. It's usually a state-by-state basis. But for sweet potato, there are certified seed programs in most major producing states. Right now, if you can buy certified seed in North Carolina, but you can't move it across state lines into other sweet potato growing regions. So
again, I was talking to Kathy the other day, and she made not the joke, but like the observation that a lot of people in the South like to buy equipment from people or farmers in the Midwest and that kind of thing, and that it was really clean equipment. It sounds like that's less of an issue here, but I'm sure you can move these things around with equipment as well.
Oh yeah, I'm sure you can too. Yeah, that's always a good, anything that can move soil can move a nematode, So the importance of cleanliness, moving between fields and between operations will always reduce the spread of soil-borne pathogens. Yeah.
Perfect. Yeah. Watch out moving these things around. It sounds like you don't want it if you're growing sweet potatoes.
Yeah, exactly.
Looking for IPM funding? Visit your regional IPM center's website.
Well, I'll start into these wrap-up questions then, Will. I mean, we kind of talked about this a little bit, but my first one for you is, what is your lab currently working on or planning to work on in the future? It sounds like it could be a lot of different things, but yeah, what are you guys doing now?
Yeah. The major push in my lab is identifying new sources of resistance in these vegetable crops. Often we're using wild germplasm that's available through the USDA germplasm collection, screening that for resistance to these different nematodes, and then trying to move it into more horticulturally favorable lines that would be useful to a breeder or to a grower depending on the crop. And so that's the main push in my lab is to produce new resistance tools, but also we have programs working on projects working on new detection methods. So we just published a paper on a new detection method for sweet potato, actually. So instead of having to speciate individual nematodes that are dissected out of a root, we're simply we've came up with a method of basically taking whole DNA from the skins of sweet potato and being able to detect Enterolobi at a low level on those sweet potato skins. So we're hoping to expand that into other sweet potato pathogens that might be becoming a problem on sweet potato seed roots as well.
Another thing I read, I think in the annual review, was like, I think one of your things was, one of the points at the end again was trying to make more international collaborations to understand root knot nematode. Like, do you know of anything that's currently being done on that front?
Yeah, I mean, there's nematologists working on root knot nematodes all around the world, and I think there are efforts ongoing to try to, we just had a meeting between our government nematologist counterparts over in Mexico and Canada just a few weeks ago, the Prasa Norte working group, to discuss possibilities for international collaboration on a lot of these nematode problems. So I think that hopefully that something will come of that, and we'll move forward, start to be a little more open about sharing information and sharing germplasm, because it's definitely needed. We're definitely not the only ones having problem with Enterolobi. Actually, was just reported this past spring in Australia in sweet potato. So that was a big deal, and hopefully maybe somewhere in Australia will hear this podcast and reach out.
Yeah, I hope so.
Because we'd love to work with them. I think there's definitely some things to be learned by how different countries have tried to contain these nematodes and information about the potential virulence and distribution, future distribution of these nematodes. So.
Yeah, probably not to mention like all the different avenues of information we have about the genomes of these nematodes. I mean, there's lots of different avenues that people can explore. I mean, like you said, you can't, one person can't study it all, right? Like, so.
No, for sure. There's a lot of questions that remain to be answered, and I think it could be answered a lot faster if we started collaborating a little more internationally. Yeah.
So my next one for you, I'll ask a multiple part question here. What do you suspect the future will hold for root knot nematode management? And then I'm going to expand it to like, what do you think it will hold? What do you think the future holds for nematode management? And then one more, and you don't have to answer this last one if you don't want to, but I'm just curious if you have any ideas. Like you mentioned, working with root knot nematode, you can learn about, the most basic ways a plant defends itself because of the success of this nematode on so many different hosts. You can almost learn. But so yeah, what do you think the future holds for the management of nematodes and root knot nematode and plant host defense, I guess?
That's a big question.
That's a lot. That's a lot. Tackle whatever part you want.
My admittedly biased answer. Yes. I think there's a lot of opportunities to improve host resistance, and with the increased use of biotechnologies and advanced breeding approaches, I think there'll be a lot more availability of resistant cultivars in different crops. I also think there's a lot of, although we lost a lot of broad spectrum nematicides like methylbromide, chlorpicrin that used to be used a lot, or relied heavily on. There's a new generation of non-fumigant nematicides that are a little more specific as well, that are coming available on the market, that I think together, though, we can come up with a good IPM, integral pest management recommendations, integrating both host resistance and nematicide based approaches. I think that's really the key. And as well as I would say, I don't know if it's a prediction, but I think in order to manage them successfully, we need a lot of grower awareness as well as to the problems these nematodes cause, because it's even with something like root knot nematodes that, like we were discussing, that are creating these really drastic changes to the root, most growers are unaware they have a problem.
Interesting.
So I think the more awareness growers have of the potential problems that nematodes can cause, not just the root knot nematodes, but there's a lot of other genre. I think root lesion nematodes, are really an understudied and huge problem that we really don't fully understand how bad of a problem they are, because they're just so cryptic in the damage they cause. So I think the future, we won't just be looking at resistance to nematode resistance and management of root knot nematodes, but be looking at resistance and management to other genre besides just cyst and root knot nematodes as well. So
yeah, that's really important. This is not the only nematode that affects plants. It's one of the most important, I'd say. But I suppose, genetically speaking, we're learning about all these other nematodes as well, not just root knot, right? So
absolutely, yeah.
Cool. Well, my final question to close out these podcasts is always this, Will. I ask people to kind of sum up, if you were going to tell a member of the public, and I've cited my mom as just somebody who doesn't know what root knot nematode is, doesn't know how nematodes are affecting crops, how genetically we're making advancements in understanding nematodes and plants and everything else. How would you just succinctly talk to a member of the general public about what your work is and why it's important?
Yeah. I would tell them I research parasites that infect plants, and I provide growers with the tools they need to provide you with better and cheaper vegetables.
Perfect. Well, with that, Will, this is the point in the podcast where you have earned your right to make acknowledgments and point people towards any resources that you might want to point them towards, or maybe just your lab. And I'll throw as much of it I'll throw what I have in the show notes for people to find. But now's your time. Go for it, Will.
Okay. Well, I'd like to thank all my collaborators at different universities across the Southeast. My unit, the U.S. Vegetable Lab there in Charleston, South Carolina, and all my collaborators, technicians, and students who work hard there. And if you would like to know more about Melodia antrolobia, I would encourage anybody to look at our project website. That's findmenematode.org. And now we talked about distribution and management approaches that could be used by growers and the public. So
perfect. I'll get it in the show notes. I'll beg Will for some photos, because I think we could get some really cool photos up on some social media. Check out Icy Dead Plants on Twitter for some of that. And
I got it written down. I'll get you some.
Great. So with that, folks, Will, thank you so much for coming on. This has been really fun, really interesting. It's gotten me energized. thinking about the future of nematode management. So
Thank you, Ed, for inviting me on. I really enjoyed it. So.
Absolutely. With that, folks, thanks for listening. We'll see you next time.
I See Dead Plants bridges the gap between plant science research and the impact it has on our daily lives. It is brought to you by the Crop Protection Network and the North Central Integrated Pest Management Center, which are both partially supported by the USDA National Institute of Food and Agriculture. For more information on today's topic or other pest management issues, check out our show notes, cropprotectionnetwork.org or ncipmc.org. Remember, this information is for entertainment purposes only. The statements made in this podcast should be interpreted within the limited context of the particular topics being discussed. Contact your state extension program for local information on pest management. For the full non-discrimination statement or for accommodation inquiries, go to cropprotectionnetwork.org/about. Thank you for listening to I See Dead Plants.